Hi, well, hello, everyone. My name is Nigel Browning, and I am Chair of Electron Microscopy at the University of Liverpool, and also the co-founder and Chief Scientific Officer for SenseAI. And today we'll talk a little bit about how the sparse sampling all fits together with Volume EM. And now over to you, Roland, if you want to give a brief introduction.
Thank you, Nigel. So I'm Roland Fleck, Professor of Ultrastructural Imaging at King's College London. I've been active in EM for many years, working in both room temperature and cryo techniques, with a background in cryopreservation and organ preservation. So, Nigel, back to yourself.
Okay, great. Okay, so Isabella, if we can have the first slide. Okay, so today we're going to talk a little bit about sparse sampling or subsampling. I thought it was a good thing to start with, just to make sure that everyone is aware of what we're actually talking about. So for all of the scanned probe or scanned electron beam techniques that are out there, most of them follow the idea of what's listed in part A over here, which is a raster scan, which is that the whole thing moves across to one side, flies back, and moves again. And this is the regular scanning that takes place inside most electron microscopes. Now, you can think about how you might randomize that scan, which is instead of having a raster scan, is to just do random points. And then you can think about, well, maybe we can actually not take all of those points, and then you can do a subsampled or a sparsely sampled random scan, where you take points as shown in part C on the slide that's here. Now, in a lot of microscopes, there are hysteresis issues associated with the scan, and so it's easier to do a random walk than it is to do a complete random scan, and that's what we have shown in this line hop scan at the end. So essentially, what we're going to be talking about today is applications of using this subsample scanning process to get faster acquisitions, shorter acquisitions, less dose, and generate less data, so it's quicker data transfer going forward. Isabella, next slide, if you can push that one on. All right, so here is just one example of that. This is a sparse sampled, actually a 4D STEM image that's taken from a Joel Grand Arm instrument. And you can see in the middle here, we have the sparse sampling of the gold nanoparticles. The dictionary that we use for the reconstruction is shown on the bottom. And on the right-hand side, you have a full atomic resolution image. Now, because of the SenseAI approach has very fast computational speeds, we can actually reconstruct this in live time. And so these is actually seeing how it would come off the microscope in terms of the sparse sampling. So effectively, this is the basis that we'll be talking about today. So, all right, so thanks for that, Isabella. You can stop that now. So that's sparse sampling. Now we want to talk a little bit about where Volume EM comes in. So Roland, if you don't mind giving us an introduction to Volume EM and your particular interest in it.
Okay, thanks, Nigel. So I'm assuming we've got quite a diverse audience, so I'll sort of build this up slowly. Any questions, put them in the chat. We're happy to answer them, provide additional information if required. But basically, Volume EM is just an offshoot of general electron microscopy. It's really come to the fore over perhaps the last 10 or so years, largely associated with improvements in the instrumentation. But what does it really mean? So essentially, electron microscopy allows you to achieve higher resolution data than you would with perhaps light microscopy, simply due to the wavelength of the imaging source. But normally we're quite limited in terms of the field of view and the thickness of the sample. So that's generally considered the biggest limitation or weakness of electron microscopy. So volume electron microscopy essentially brings together a group of different techniques which allow you to generate electron microscopy level resolution, but over large X, Y, Z volumes. And we achieve this normally, not exclusively, but normally using scanning electron microscopes, where the sample is loaded into the microscope and then imaged using the scanning electron beam. There's essentially 3 different flavors of this. One where you section your material conventionally using an ultramicrotome, collect your sections on a suitable substrate, something like a silica wafer, and then load that into your scanning electron microscope, or the ones where the microscope itself has been modified in some way to allow it to cut 3-dimensional volumes. So you've got 2 options here. One where the microtome's introduced into the chamber, so that's serial block-based scanning electron microscopy, SBF-SEM on the diagram, or vEM, where you have a focused beam of gallium or plasma, which is actually used to sputter away a controlled thickness of the material, exposing a fresh layer which can then be imaged, and then you repeat that by sputtering away yet another layer and then re-imaging it, and with this you can build up a sequential series of data sets which you can then in silico stick back together and generate your three-dimensional volume. So this slide, the orange section, really highlights the common or established techniques for volume EM. They're all room temperature, relying on chemical fixation, some form of dehydration, chemical staining of the sample, resin infiltration, and embedding. But one of the areas I've been working on for the last 3 or 4 years, and actually it established the collaboration with Nigel and the guys at Liverpool University, is to actually do volume EM in the cryogenic state, where actually we use high pressure freezing and plunge freezing to vitrify the sample in this way that is essentially perfectly preserved in its native biological state, and then we image it inside a cryo-vEM and you sequentially build up a large number of sections and then reassemble them in silico. So all of these techniques are quite challenging. They're slow. So anything which improves throughput, so sparse sampling, which Nigel highlighted in the previous slide, offers a benefit to us. In the cases of the FIBs, where you're using plasma or gallium, these sources come with quite a high charge cost associated with them. So anything which allows you to generate volumes quicker and with less consumption of your gallium or plasma source is probably advantageous. And that's really where we've been working for the last few years. Nigel, do you want to take over for a bit and
Yeah, no, thanks very much for the introduction into volume EM, and you've sort of highlighted some of the issues in terms of the throughput. I wonder if there are other things that are challenged as well in the actual imaging process, or things related to the quality of the images that come out. Or what are the major challenges that are out there in terms of the resolution and the speed of what you can generate with the technique?
Okay, so a lot of the challenges just come down to the sensitivity of the sample itself to beam damage. That's particularly prevalent in the cryo environment, but also exists in the room temperature environment. A lot of your preferred sample preparation approaches, your high pressure freezing, freeze substitution into resins tend not to be compatible with techniques like serial block face scanning electron microscopy, because the resins are quite sensitive to beam damage, and this makes it a little bit limiting. In the case of the cryo environment, they're extremely sensitive to beam damage. Basically, you can devitrify or melt your sample in the crudest sense. And all of these techniques in the scanning electron microscope are quite prone to charge accumulation. So as you hit different parts of the sample, sometimes you get an accumulation of charge, which causes a distortion in the beam and just basically detracts away from the quality of your data sets. So again, something where we've worked closely over the past few years to try and exploit the line hop random scanning and sparse scanning approaches.
Okay, great, thanks. And so what do you think are where could volume EM go? Is there some examples you can talk about of what you could do and what you could work on if you could get beyond some of these throughput issues or charging issues in terms of the imaging side of things?
So for the room temperature community, it's really been led by the neuroscientists. They have a big push to do whole brain, volume EM, highly challenging, massive scale in terms of the size of the block that they're trying to actually section. You see people at Jamelia Farm parallelizing instruments so they can run multiple blocks at the same time in parallel. And they discuss numbers in the regions of sort of you at least a year, if not years, of data acquisition collection, petabytes of data, and you probably even longer assigned to post-processing of the data. So as an example, if you could scan an image in say a quarter of the time, if we're saying 12 months as an off the top of my head number, you'd be doing this in a quarter of a year instead. So there's a big gain. It's not just time, there's just a risk factor. Trying to keep a facility, a lab stable for 12 months or longer with absolutely no interruption to anything is quite a challenge. You know, the things happen, you have power outages, you know, unforeseen events, even just a problem on the highway can stop staff coming in to maintain an instrument. So the shorter this acquisition period, the better it is for the end user.
Great, thanks. And do you want to just show an example of how you've used the sparse sampling to get around some of these issues?
I think we have that coming up in a few more slides. Here we go. Perfect. Okay, so this is a Euglena gracilis. It's a flagellate euglenoid. This is vitrified, so this is a unstained. It was living and swimming at the point of vitrification. I have actually frozen them and brought them back to life again. So in principle, this is a fully viable cell. The images on your left-hand side, I assume it's mirroring correctly for the audience, are a raster scanned image, and the charge accumulation, the dark areas and the patches and the streaking are more prevalent on that side than they are on the right-hand side, where we're using an interleaved line hop scan and subsampling the data. So by having a more stable charge environment, it makes the post-processing of the data easier to manage. We are kind of using a semi randomized scan approach on our raster frame. What we do is we generally accumulate frame integrations. So we do a fairly fast scan across the block face, and we do this multiple times and then reassemble each frame to create a single image, and that's actually what you're seeing running here. But this is not as efficient as using a random scanning pattern across the surface. Nigel?
Okay, great, thanks. And so where do you think this will go in the end? What's the dream of the process that we use?
So the dream's fairly straightforward. If I can vitrify everything, you know, single cells, blocks of tissue using high pressure freezing, for example, and then volume image them in the cryogenic state. The organization of the cells, their organelle, is really as it was at the point of freezing. So it's as close to perfect as you can get. This makes it much easier to interpret the data. If you're doing fluorescence clem, the fluorescence is preserved. You can identify regions of interest for targeting, for sectioning. And in principle, you can do quantitative analysis on your data sets. And we've managed to do this with a Chlorella vulgaris study, where the group we were working with were mutating it to change its color. They were intending to reduce its reliance on photosynthetic growth and move it to a heterotrophic growth condition, and in turn change the lipid droplet concentration within the cells. And Volume EM proved to be extremely efficient and effective at quantitatively measuring these changes during mutation.
Okay, great. Well, thank you very much for that. I think that's sort of what we were aiming for in terms of an introduction to how sparse sampling can fit into Volume EM. Now, if anyone has any questions, then both Roland and myself are quite happy to answer those questions.
Okay, so let's start out with do you have any other examples of experiments or data that we can see here today?
Possibly not loaded up as slides. Nigel? Okay, here we have one. So this is an assembled one. So actually the frame integrations, which you saw in the previous slide, that was essentially one slice from a volume where you were looking at either the frame integration or multiple frame integrations acquired using line hop. This is after that process, where essentially you've got your 50 frames reassembled as a single slice, looking at the raster image on the left and the line hop data set on the right. And if you look at the left hand one where the little orange arrows are pointing, And if we could replay that, it'd be really useful. You'll see the Palladium bodies actually start to decay and break down due to beam-induced damage, which doesn't happen with the line hop sparse sampling strategy.
Okay, great. And then in general, I think we only brought examples here to show of the volume EM. If anyone's interested in examples for other sides of it, it's all on the SenseAI website. You can see everything from STEM through to SEM, EBSD, EDS, and other examples as well.
Okay, next question. What does it take to install this sparse sampling technology on a microscope?
Okay, Roland, do you want to talk about specifically how you did it, and then I can talk a little bit more about in general.
So we have a external scan controller, which gives us the scan control necessary to implement your line hop solution. There's a large PC running the SenseAI software. It has all the control parameters, and it manages the percentage of subsampling. So quite often we'll be running at 25% subsampling. We have gone down to 12.5%, and then everything afterwards is then done on that software suite. So the dictionary learning and reconstruction. Nigel, we maybe want to go over the dictionary learning strategy.
Yeah, so let me just take in general the bit about where the installation comes from and how do we do it. Essentially, the SenseAI solution is a plug and play way of going through it. We plug into the external scan port on any microscope and basically use our software to take over control of generating the scan. The output from whichever detector you want to use then comes back into the software, and we reconstruct the images. And so effectively, the process takes about half a day to install any microscope. You can put it together, you go through the process, and then we use, as Roland said, dictionary learning to do the reconstructions. And so this is a way of essentially taking the sparsely sampled data sets, understanding what the components to those images are, and then reconstructing them into the full image going forward. And there are various things online. I think previous webinars have shown how to do that, and then there's all kinds of different ways to go through that and apply the mathematics to get it to work out.
Okay. Let's see what other questions we have. Does installation affect any techniques on the microscope?
No, it's basically it just sits there entirely passively. If you don't turn on the scan generator, the microscope's exactly the same as it was the day beforehand. Yeah, if you don't launch the software, it's just a standard SEM, yeah, or FibSem or serial plot face SEM or whatever.
Yeah, I mean, basically the idea is, I mean, it's just like having a scan unit on a microscope. If you don't turn it on, it doesn't affect anything else that you're doing. Also, anything that we do with the SenseAI software doesn't have any effect on the physics of the scattering process. It doesn't change the way a detector works. It doesn't change the way the microscope operates. It's really just taking the output from the instrument in a different way to get the sparse sampling and then putting it back together. And that means whatever the performance of the detector you're using, whatever the performance of the technique that goes into that, you're effectively just speeding up that by typically a factor of between 5 and 10 over what you get from a normal detector or the normal use of a particular method on your microscope.
Okay, looks like we're getting a few more questions here. Let's see. So someone asked, how do you decide this 25% or 12% or whatever percent in subsampling?
So I'll do a quick one, Nigel, then I'm going to throw it to you. So, the obvious is, I'm an academic end user, so essentially, I don't develop the code or implement or anything like that. So we were trying to resolve a problem with charging and everything else. So we've tried interleave, we've tried align hop, we use frame integration. You could potentially beam blank as well. But the real advantage is this subsampling, taking a smaller percentage of the whole image and then inpainting it. So with the tests we did with the team in Liverpool on the academic side, for biological tissue, it seems that the lower threshold's about 10%, from 100 down to 10%. When they compare 100% image to a 10% image, there's basically no difference. 12.5, 25%, they're essentially identical. When you start going below 10%, the samples just aren't sparse enough for the dictionary learning to work effectively. Now, I have been told if you spend enough time, it will work. But Nigel, maybe you want to explain that better from a more mathematical standpoint.
I'm not sure there'll be that much more math involved in it, but the idea is that for all of the applications that we've done using Sensei and going through the sparse sampling, effectively you can get a lossless reconstruction back to the original data set if you're anywhere down to 10%. So the challenge, of course, is to do how do you verify that? Now, the way that we have verified it in the past is that we have the ground truth 100% acquired data for areas where we don't have beam damage going into the system, and then we subsample that data and we do a reconstruction and compare the original 100% image compared to the 10% sampled image, and that will tell you how accurately you've done the reconstruction. So when you have conditions that you can do that with, which are the cases where you don't have charging, you don't have beam damage, you don't have any issues with the acquisition, then we have found that essentially anything above 10% gives you a very good reconstruction. Anything around 20%, you're absolutely guaranteed to get a lossless reconstruction that comes out of that. Now, that doesn't mean that that's the only level of subsampling you can do. It just means that that's the one that you can do without having to worry about how accurate the reconstruction is going to be. Testing it on places where we have beam damage or where we have charging, of course, is impossible to do, because if you could get the image without charging or without beam damage, you would never want to use the sparse sampling. The sparse sampling is correcting for an effect of the beam. So we don't have the ground truth in those cases. And so we tend to go with just the ones that we've known work very well in every single case that we've looked at. But as Roland was saying, this is a way of balancing. And so essentially by having the sparse sampling, you have another degree of freedom in the way that you balance the electron beam that goes into the sample. And so that means that you have another knob to play with, another way of looking at it, another way of balancing magnification with sampling, with process and time, dwell time, to get the right answer to come out. In cases where we haven't had to worry so much about getting back to the original resolution precisely, for example, we've done some things on some NASA data sets from satellites. In those cases, we can get down to about 0.5% subsampling and still reconstruct the image with the accuracy needed to solve the problem. And for microscopy, what that would be is to say that, if you don't want the 1 nanometer or 1 angstrom resolution of the microscope, and you can maybe go to 2 nanometers or 2 angstroms, then you can do more subsampling to go into that process. So the numbers we pick are usually the ones at the moment that we know will give us the best reconstruction for the least amount of effort.
Thank you very much, Nigel. Next question, when you average the frames collected from a single face, is the line hop pattern exactly the same for each frame collected?
Depends how stable your instrument is and how good your scan coils are. If you really want to push, if we're doing frame integration, because we do use the same external scan generator for that as well, we will motion correct the individual frames before assembly. Nigel?
Yeah, so the system itself, the actual sparse sampling system, doesn't have to have exactly the same subsampling from one frame to the next. Obviously, if you're going to try and use some interleaved method where you're going to use things that are using multiple frames that are subsampled together, then it helps if you're balancing those out against each other and the pixels that you're going to sample. But in principle, we can go through and we can use a different subsampling pattern for every frame that we are acquiring do the reconstruction from the subsample data, and then average those together in the same way as you would average any group of images that you want to put together. So yes, you can do it. Don't have to. It's really, again, what I was saying before, is a degree of freedom that we can use to help solve the problem that we're trying to address.
Okay, next question is pretty straightforward. Can this be implemented for lamella preparation for cryo-ET in a cryo-vEM?
There's no reason not to. On the other hand, I tend not to look at the lamella particularly closely with the vEM, really from the perspective of trying to reduce any beam damage or beam interaction on the sample before I get to the TEM. Yeah, yes, no reason why you couldn't. If you want to have a good look at the lamella and you want to minimize the damage going in, then yeah, it'd be the same thing. In fact, it'd probably be only one 2D image that you'd actually want to reconstruct as well, which would make Nigel's life a lot easier than my volumes.
Yeah, so I'll just say the same thing. Basically, is that you can use it for imaging in any way that you want to do it, and any way you choose to bring the sample out. For the examples that Roland is showing here, is just blasting away one layer and going to the next layer. But if you wanted to cut that layer out and then get a reasonably high resolution image from the SEM before using that to cryo-ET, then you can do it. It will have a lower dose. Obviously, you'd have to understand the full dose fractionation and how much dose you go into it, and everything you put into it inside of the SEM, you're still going to have to worry about that for the tomography experiment. But because we can do much lower doses in terms of the process, then it should be easy to do that. Now, of course, it would also apply the subsampling to cryo-ET as well. So you could do lower doses on those and get the same information to come out. So the distributing the dose can work in multiple different methods rather than just from one in terms of the process that we're using.
Okay, thank you both. One more question here, and then to the audience, if you have any other questions, please make sure that you enter those into the questions tab in the bottom right-hand corner of your screen. This person wants to know, what is the hardware setup at King's College that you're working on, Roland?
So I'm using a Joule 4700 vEM, equipped with a Leica VCT500 cryostage cryo anti-contaminator. That's supported by the Leica VCM cryo loading station, Leica ACE900 vacuum transfer and coating capability. And then on top of that, we have the quantum detector scan generator and a standard Dell gaming computer with the SenseAI software stuck on it, used for data acquisition and not for gaming, I might add.
Okay. Well, looks like we are coming. Oh, just kidding. Another question. What other volume EM applications have you got lined up for sparse sampling at King's College?
Well, we do array tomography. We have serial block face imaging, both of which we have run basic tests on SenseAI software. It does what it's necessary to. Generally, we don't suffer too much from charging on those types of samples, and they've been stable, and the projects have been running for a fairly substantial period of time, predating this project. Let's put it that way. So we've not changed the established protocol. We would consider it for new projects. Most of the focus of the last 2 or 3 years has been Marina's work on the cryo volume imaging, where we've really worked extensively with Nigel and the guys in Liverpool to try and improve our signal to noise. These are vitrified samples. There's no metal contrasting in them. They're really beam sensitive. So we've done an awful lot of work in this area to allow us to work robustly and stably for extended periods of time. Joel have also invested a lot in developing software to allow us to track the face over long periods, and we can run for multiple days at a time now, acquiring sections. So SenseAI has just basically allowed us to get 4 times more data for a given period of time on what is a very, very stable platform. You know, 7 to 9 days of data acquisition would be considered fairly routine now.
Wow. Okay, next question. What dose is being applied to the sample with the line hop method compared to the raster method during image acquisition?
I'll give that to Nigel because you know the calculations.
I mean, essentially, the way that we usually do it is just a fraction of the pixels that you're acquiring. So if you're 25%, it will be 25% of the total electron density that goes into the sample compared to the original one. Now you can start thinking about balancing that dose out in terms of, would you use the same dose that you did for a raster scan for a subsample scan, or would you increase the dose per pixel, but then you have fewer pixels? And so this goes back to the case of how having that extra degree of freedom to go through the system. So if you're only using 25% of the pixels, you might increase the electron dose in each pixel because the integrated density going into the whole sample is going to be lower. If it's a charging effect, then that means that's going to be less of a charging effect, and you can increase the electron density to get better images in that way. If it's a damage effect, there's still a balance that goes into that, because in a raster and doing a raster, you effectively have a cumulative damage as you move along in that process. And if you're jumping into different areas, you're doing the line hop method to go through that with the random walk, then you're distributing the dose over a larger area. You're also going to that point of not accumulating it as rapidly. So you get things of a benefit with the total dose, you get a benefit with the dose rate, and you also get a benefit from the dose overlap. And so the simple calculation is just number of pixels you take versus the number of pixels you would have to start with. But in terms of the overall damage or charging effects on the sample, it's a little bit more complicated to do that. And that's why we have essentially within the software a way to do a live imaging technique so that you can balance all of these things together to create the best images that you're acquiring while you're sitting on the microscope. That's a key part of being able to get live images. It means you can balance everything with the microscope when you're on the microscope doing the live images, so you can get it right. Then you take your section and then you move on and go through with that. Because essentially what we all want for any application, whether it's material science or it's in structural biology, is you want to put as many electrons into the system as you possibly can without causing a deleterious effect, because that's going to give you the best image coming out of that. And that's what we can do with the Sensei system.
Okay, thank you, Nigel. I think now might be a good time to ask the audience, if you're interested in a demo, would you like to see more of this? Do you want to talk to an application scientist on the team? Maybe you have some questions or concerns. We'd love to follow up with you. So if any of these options apply to you, we'd love to touch base after this webinar is over. And we can allow for a little bit of time for anyone to answer or to ask any remaining questions that they might have. Okay, Roland, Nigel, any closing thoughts that you might have for the audience today?
No, I've been very happy with the installation and the collaboration with the team in Liverpool. It's worked well. It's certainly giving us nice results, and yeah, the dictionary learning, it's an interesting and positive direction of travel for us. Nigel?
Yeah, I think to add to that is that we can implement this on any combination of instrument, hardware, detector it doesn't really matter in the process to go through with it. So effectively, as I said before, we don't really change the physics of the process. We don't change anything related to the hardware that's involved, and so it can be applied to any system. It's really just a software fix to the problem of getting too much beam into the sample and getting the right amount of image data coming out that allows you to get the most information from the experiment that you're doing. So we're very interested in hearing from really any combination of hardware to get this to work out.
Okay, well, thank you everyone for your attention and your time. Thank you for the great questions. And thank you, Nigel and Roland, very much for your time today.